So I guess the first question is, why do we want to talk about genes today? Well, I think, in general terms, the study of genes often provides us with some insight into the disease mechanisms. So, why a disease develops, what are the steps and processes involved, and ultimately how can we try and kind of intervene and stop or treat a disease. I guess the other reason for talking about genes is that a lot of patients and their family members worry about passing on a disease like FTD. And, it's, I think, important to try and address some of those issues today. So if we look at sort of disease mechanisms first, and this is kind of a bit of background cell biology. Maybe this is familiar for you, maybe it's not. But I thought it would be helpful just to have a bit of an overview. So, functions within a cell are performed by many, many, many different proteins. Now, some of these proteins convert some molecules or even other proteins from one form to another. Some of these proteins support the cell structure, and some of these proteins transport genetic material around the cell for processing or building other proteins or whatever. And the blueprint for each of these proteins is a separate gene. So there's a little part of the DNA sequence that maps out for this particular protein, and that's the crucial bit if you want to make protein X. Now, if you have an error in that piece of the genetic material, an error in that gene, or what we call a mutation, then the resultant protein might be a different size or a different shape, or one of its crucial functions may not work anymore. And this can have devastating consequences on a cell. It could be toxic to a cell, or you could just lose a vital function that the cell needs. So in this sense, understanding the study of genes helps us understand what has caused a disease in a particular person who carries that particular gene mutation, but it also gives us an idea of how that disease may develop. If we understand, okay, this protein, when that's abnormal, the disease results, then we could say, what does this protein do, and why is that important for the development of FTD or whatever genetic disease we're talking about? Now, I'm sure you can understand that this is an unbelievably sophisticated and complicated system. I mean, we're talking about hundreds and hundreds of thousands of different proteins that interact in either just one-to-one or one to many different other proteins. So it's a very, very complex and difficult issue to get our head around. And that's obviously why it takes so long and so much effort to try and understand. One of the first ideas I want to introduce is this idea of, are we talking about one gene or many genes? So if the disease is caused by just one single faulty gene, just one gene is abnormal and that's enough to cause a disease, we call this a kind of monogenic. So mono meaning one gene, a monogenic disorder. Now, if this is a very uncommon situation in FTD, and I'll come to that in a little bit, but perhaps only 5% of patients have disease caused by one single faulty gene. Now, if you're unfortunate enough to carry one of these particular genes, then there is a significant risk of passing that on to your family, unfortunately. Perhaps a more common scenario is that there are a number of different genes that interact together, perhaps, or also with other environmental factors. And in this situation, we call this a kind of many genes, polygenic type of disorder. Now, this, because we're talking about a mix of genes and a mix of genes plus environmental interactions, the risk for immediate family is probably less than the monogenic form. But it may still be a bit more than the general population. Now, I'm going to come to these in a bit more detail. So as I say, about 5 to 10% of patients with frontotemporal dementia have a disorder caused by a single faulty gene. And these people often have a very, very strong family history. We'll come to exactly what a strong family history means in a minute. But essentially, if you have lots and lots of people affected in the family, then that's a bit of a tip-off for us. Now, especially with the new development, which John mentioned, we have now got genetic tests that explain the majority of cases due to a single faulty gene. So of this 5 to 10%, we can explain, probably 3/4 of those cases. So that's kind of a double-edged sword, because at least you can find out if your disease in your loved one is caused by a particular gene, then you can test it in other family members if they choose to do so, and we at least have some idea of what to expect. Now, polygenic disorders, I guess the reason for bringing up this is that there's probably 30 to 40% of people with FTD have some sort of family history. Not the very strong family history where there's 2 or 3 members of each generation that are affected, but there may have been a great uncle that had a young onset dementia, or there was somebody else had some sort of abnormal behaviour that was perhaps diagnosed with a psychiatric illness, or somebody else had Parkinson's disease. So not a really kind of obvious and strong family history, but something there lurking in the background. And I guess the question is whether this is due to a combination of genes predisposing members of that family to frontotemporal dementia and related diseases. So I guess the question is, what constitutes a family history? So these are the sort of things we ask about when we're asking about family history. Obviously, we want to know about late onset dementia, but actually, late onset dementia in the family, that is, a family member who developed dementia in their 70s, 80s or 90s, that's actually very common. That's most people, or maybe perhaps not most, but a significant proportion of the population have that kind of family history. So that's very common. But we're also particularly interested in people who have a family history of young onset dementia. So people that have dementia that begins, before they hit the age of 65. We also ask about a history of motor neuron disease, and that'll become clear. The reason for that will become clear in a moment. And then I've put here this phrase I've come up with, poorly recognised illnesses. So, this is an evolving field. We don't always, I guess what we know now is not the same as people knew even 10 years ago, or 15 years ago, 20 years ago. And so there are all sorts of kind of funny illnesses that we're never quite sure about, family members that just seem to lose contact with other people and we don't know where they ended up, or, some sort of bizarre behavioural problem ended up in an asylum, 50, 60 years ago. There's all sorts of kind of funny family history that makes us wonder. Now, this is the fried egg of genetics. This is a pretty common way of thinking about neurological diseases, I guess any disease, and that is that you think of a very small proportion of disease being due to faulty genes, and a bigger proportion of disease being kind of due to chance, if you like, and they use this term sporadic. But if we just put a new label on this and say, well, perhaps this group are the monogenic ones. So this is a very small proportion of people with frontotemporal dementia that have disease caused by a single faulty gene. And then really, if you think about sporadic disease, perhaps that's actually polygenic, right? So that you may have 10, 15, 20, 30 genes acting in combination with some sort of environmental factor which leads to disease. In a sense, when you're doing research, the families that have kind of very, very strong family histories where there's multiple affected individuals, they kind of stick out like a sore thumb. And the research is not easy to do, but it's certainly a lot easier to do to study a family like that and to find the faulty gene. And once you find a faulty gene, you can test it in other people and you get a much clearer idea of what's going on. I'm sure you can appreciate just from what I've said, if you're talking about who knows how many different genes and who knows how many different environmental factors and who knows how many different interactions between all of those, this area is unbelievably complicated. So of these two situations, we know much more about this actually than we do about this. We know a lot about the rare and not that much about the common. And that will change over time, but this is a very, very challenging kind of situation to deal with. I want to just introduce another concept here for reasons that will become clear in a moment. But one of the weird things about frontotemporal dementia is that sometimes it seems to kind of run in kind of family clusters, if you like. So what I mean by a family cluster is that, there may be FTD in one family member, or perhaps motor neuron disease in another family member, and then a third member of the family may have a combination of the two. So in other words, a kind of variable disease presentation and a variable inheritance pattern. So if we represent this kind of graphically, this is a way that we sort of think about family history. So just to orientate you, the circles are women, the squares are men. I'm not sure what that says about blokes, but anyway. If you see a line connecting the two, that's a partnership where they've produced an offspring. And so in this situation, we've got here, grandparents, parents. Well, hang on, that's probably not too many. Great grandparents, grandparents, parents, and children, for example. So each kind of level down as another generation, and then these are all their ages, and if you've got a line through an individual on an angle like that, it means they've passed away. And then if you colour in a circle, usually, or colour in a square, that usually means they have the disease you're interested in. So in this particular example, what they've done is they've shown a history of frontotemporal dementia, FTD, as being a coloured in bit on the left of the diagram. So you can see that there's someone with FTD there, and there, and there, and there, and there. They've also coloured in people that have a history of motor neuron disease. So on the right side, so there's one there, there's another one there, and then they've coloured in people who have a combination of frontotemporal dementia and motor neuron disease as having a full black circle or square. So you can see this is kind of a family cluster. Now, it is still a pretty strong family history in the sense that there's an individual affected, at least one individual affected in each generation. But, if you're talking about FTD in one family and, MND in another, the connection may not be that obvious initially. But, this is an area of significant research over the last couple of years. And really, this is the major breakthrough of the last couple of years, is that there were two papers published in the same edition of a journal, a scientific journal, both in the same issue in September, I think, perhaps August of 2011. And this is this thing called the C9ORF72. Fantastic name, rolls off the tongue. Some people call it CORF, some people call it C9. You can call it whatever you like. If you remember it, you'll be doing well. But this is really a kind of a new cause for FTD and motor neuron disease and the combination, FTD-MND, only identified in 2011, so very, very early days, really. Now, I won't tell you about the kind of nitty-gritty of this particular gene because I don't think that's relevant to today's discussion, but it is a kind of different type of genetic lesion which may have some implications, but it's something that's again a sort of intense subject of study. So this gene actually does seem to explain this funny family cluster situation that I've been talking about. And it's an important cause of what we call familial. So that's those kind of single faulty gene, runs in families, FTD. And it's also a very important cause of familial motor neuron disease. But interestingly, it can also be found in people that don't have a family history. So that's, again, in a significant proportion of patients, actually. So this is something that we need to keep in mind when we're seeing people with FTD. So who seems to carry this gene? How can we tell that they might have the gene? Well, the combination of frontotemporal dementia, FTD and MND seems to be a big clue. So if you took just people who had that combination, the family history, with a positive family history, perhaps as many as 40% of patients may actually carry this gene. So that's almost half. I mean, that's a really big number. If we look just at FTD, and you looked at all people who had FTD, probably somewhere between 7 and 12%. It varies a lot from one study to another. Some studies say a few more, some studies say a few less. And if you just concentrated on those people that had a very strong family history, then perhaps 10 to 15% would have this mutation. And we get sort of similar numbers for MND, but perhaps slightly bigger proportion of MND, familial MND cases might be due to this repeat expansion. So you can see already that it depends a bit on how the person presents, what problems do they have, will influence how likely the diagnosis of this C9 is. So looking at sort of, again, repetition is good here, clues to a genetic diagnosis or genetic cause of FTD. So a strong family history. So what we typically look for is at least 2 or more first degree relatives. So that's, a father or, a sibling who have either FTD or MND. I think that's pretty clearly a strong family history that would kind of raise our awareness. The combination of FTD, MND, for obvious reasons, would definitely make us think about a genetic cause. And then sometimes there can be unusual symptoms that might tip us off. So, here we're particularly looking at what are called delusions. So this is where you get the patient may get a funny feeling or funny idea about how things are working. They may be worried that the neighbours are trying to, steal something from them or hurt them in some way, or, they may have, a kind of an abnormal and fixed idea about how things are working, and you can't really talk them around. So that's a delusion. And actually in FTD that's pretty uncommon. So again, it sticks out when it's there, but it does seem to be a pretty strong marker for this C9 mutation. So if you happen to have delusions and you have FTD, the chance of you carrying this C9 goes up quite significantly. And the other issue here I've said is pattern of progression. So this is a bit variable depending on the gene that we're talking about. So there are some genetic syndromes that seem to be kind of slowly progressive, and there are others that seem to, if you have it caused by a faulty gene, then the disease tends to deteriorate much faster. So it depends a little bit, but the pattern of progression is sometimes a clue. So just changing gears now, let's talk about pathology. Now, we're going to hear more from Lauren about pathology, but just to give you, and we've obviously heard a little bit already from John, but again, this idea of studying the pathology, why do we bother? Well, it's another way of giving us an insight into the pathogenesis of the disease. So this is the how and why a disease develops. So one aspect is to look at the genes and see what proteins are involved, what are their functions, how do they lead to disease if they're abnormal. Similarly, what proteins are building up, what's happening when the cell's dying, how can we study that? So getting an insight into pathogenesis is important. And the idea is, again, as I've said, if we understand what's happening in the brain in the course of the disease developing, that hopefully will lead to some sort of effective treatment. If we understand what's happening, the idea is that we should be able to then intervene. That's obviously something we're all working towards. And I guess the other aspect of studying pathology is that it might help us develop biomarkers. Now, what's a biomarker, you say? Well, it may be a blood test or a scan result or some other sort of test that you can do on somebody when they're well, and that picks up, this person has FTD or whatever disease you're interested in. So if we had effective biomarkers, then that may allow us to develop a kind of early and accurate diagnosis, which is going to be important if we have an effective treatment, because you want to use it early, if at all possible. So these are just, again, some pretty pictures of this TDP43 pathology. They're pretty when you look at them on a slide, but I guess you don't really want them happening in your brain. But they do look nice. And there's another one here, fuzz, which we don't need to talk about too much. And there are all sorts of different sort of characteristics. They look there's different patterns and different parts of the brain seem to be affected. And the more you kind of study these things, the more complex you realise things are. Now, I guess, John's alluded to this in a way with this kind of clinico-pathological correlation. So what does that mean? Well, it means that when somebody presents with, for example, semantic dementia, we have a pretty clear idea of what the pathology is in the brain, even without actually looking at that person's brain. So that's a kind of very helpful clinico-pathological correlation. But the challenge we face is that not all of the types of presentations we're dealing with map so nicely to pathology. So, sometimes you can have similar clinical symptoms. So when we see somebody and there's a change in behaviour or personality and they're having this problem and that problem, then sometimes it's hard for us to decipher which of the kind of pathologies might be causing that. In the opposite direction, sometimes people can have the same pathology when we look at the brain under the microscope, but then when we look back and see what symptoms they had, actually the symptoms were very different. So this is a really challenging situation with all these diseases, unfortunately. Again, biomarkers might help us to kind of meet this challenge. So whether it's an imaging biomarker or a blood test or some other biomarker, this is obviously a major area of research for us. And again, Olivier is going to talk a bit more about future drug treatments. I guess just to make the point that the most common form of dementia is Alzheimer's disease, and Alzheimer's disease, as John mentioned, is characterised by this build-up of a protein called amyloid. But there is actually also the other protein tau, which is important for FTD, and I think, this is a big commercial interest for drug companies, and so they fund a lot of research into trying to find a cure for Alzheimer's disease, and I guess you can understand why. People often find that a bit disappointing, when you've got somebody that doesn't have Alzheimer's disease but needs help anyway. But I think, the good news here is that there's often offshoots. You might find that there's a common pathway that unites the two diseases, or perhaps a treatment designed to target the tau in Alzheimer's disease might also help with FTD. So I don't think this is necessarily such a bad thing, but it does highlight the need for an early and accurate kind of molecular diagnosis, getting to the nitty-gritty of what's actually happening. So I'm going to finish there. Just to summarise, a very strong inheritance of FTD is uncommon, but we don't really understand these interactions between multiple genes and environmental factors very well. We think of at least 2 first-degree relatives and young onset disease as being a strong family history, and studying genes and pathology might lead to a deeper understanding of the disease, which hopefully will lead to a meaningful treatment for us. So I'll finish there. And I just want to thank all the patients and families that have been involved in our research, because we can't do it without you. So thank you very much.